This is Biology 105, Chapter 2, Part 5 on enzymes. So in our last segment, we talked about proteins, which is one of the 4 classes of biological molecules. Now, when we talk about proteins, one of the most important roles that proteins can play in a living thing is that they act as enzymes. Enzymes are biological catalysts, which means that they speed up chemical reactions. And so when we look at living things, we know that the sum total of all chemical reactions that occur in that organism or in that cell is called its metabolism. So that includes all of the reactions where we bring food in and we break it down into smaller parts to pull out the energy, but it also includes the reactions that take place in order to build molecules like proteins, lipids, carbohydrates for storage, all nucleic acids, all of the various roles that we see our cells perform in order to both build and break down complex molecules. So the enzyme speeds up those chemical reactions, and it's not changed itself in the process. So most metabolic pathways occur in a series of chemical reactions, and those chemical reactions occur with the help of an enzyme. Now, the way that enzymes speed up chemical reactions is by lowering the activation energy. So we can think of the activation energy as that energy that's necessary in order to get the chemical reaction started. And so if you think about it, what we're actually doing in a chemical reaction is we're taking matter that is present in one form, and we're changing it into another form. And so I liken this to, again, playing with Lego blocks. So remember when we talked about matter, we said that the individual units of matter, the atoms, are similar to individual Lego blocks. If we can start to take one structure apart, so for example, if I build a house with my Legos, I can take that house apart and then use it to build smaller structures, maybe a car and a person, or maybe 2 smaller houses, or whatever the case may be. And so all I'm doing is rearranging those Lego blocks that were there in that original structure into something else. And that's exactly what we're doing when we perform a chemical reaction, is that we're rearranging matter. And when this happens, we have to break bonds in the original or starting material in order to produce the right conditions for that change in matter to occur. And that initial breaking of bonds requires energy input, and that's where our activation energy is required. And so what enzymes do is they make those reactions more likely to occur on their own, and there are a number of ways that they do that, but they essentially lower that activation energy bar, lower the amount of energy that has to be input in order for that chemical reaction to occur. Now, each enzyme is going to interact with one specific target molecule, which is called its substrate. So we can see in this diagram, we're looking at the lactase enzyme, and lactase is responsible for breaking down the sugar lactose. This is a sugar that's found in milk products. So someone who is lactose intolerant is unable to break down that lactose because they do not make the enzyme lactase. All of the supplements that we add to our diet when we're lactose intolerant are actually enzyme supplements in the form of lactase that help us to break that lactose down. Now we can see here that in our lactase enzyme there's a region called the active site. And this is where our substrate, or the reactant in this chemical reaction, is going to bind to the enzyme. So the substrate binds to the enzyme at the active site, and we now have something called an enzyme substrate complex. So they fit together, and the enzyme performs the chemical reaction. In this case, we're breaking lactose down into 2 smaller sugars called glucose and galactose. And those are the products that get released by the enzyme in this chemical reaction. Now, one thing that you'll notice is that now that the enzyme has performed its job, it is able to accept another substrate. It's not changed or modified or damaged in the course of this chemical reaction. So, an inhibitor is going to be a molecule that prevents the enzyme from doing its job. So it inhibits the chemical reaction. So we can see here, this is an example of a competitive inhibitor. Competitive inhibitors work by binding to the active site and blocking the active site so that the substrate can no longer bind. And so you can see here, this competitive inhibitor has a region that is similar in shape to the substrate, which allows it to sit in the active site. So now the substrate is unable to sit there in that active site, so the enzyme's unable to break that substrate down or to perform that chemical reaction, whatever it may be. And as a result, we don't get any accumulation of the product. So competitive inhibitors bind to the active site and compete with the substrate for binding to that active site so that that reaction does not proceed. We can also see non-competitive inhibitors. Non-competitive inhibitors do not bind to the active site. However, these inhibitors bind to another region on the enzyme. This is something called the allosteric site, but it's just another site on that enzyme. Okay. Now, what happens is that when that non-competitive inhibitor binds to this other site, it changes the shape of the active site. So you can notice a change from our active enzyme to our now inhibited enzyme. It's no longer able to bind to that substrate. And so that substrate, because it cannot bind, will not get broken down, and the enzyme is unable to do its job. Therefore, it has been inhibited. And then we see again that there is no accumulation of product as a result. So, like all other proteins that we've talked about, form fits function, right? So the function of the enzyme depends on the shape that it takes on. And if we see a change in the shape of that enzyme, it can change the function or prevent the function of the enzyme. So we know that lactase, we've just used in our examples, is an enzyme that breaks down lactose or milk sugar. And if we see mutations in the lactase enzyme, it can change the shape of the lactase and make it ineffective, leading to lactose intolerance. So here's our lactase enzyme. Here's the active site where lactose binds. If we have a change in shape of this enzyme, lactose will no longer be able to bind, and therefore lactase will have lost its function, and we will not be able to break down the lactose. So that is the end of our discussion of enzymes, and this is the end of Module 1 as well. In our next unit, we'll be looking at energy and metabolism.